and a PhD student at Songjun Park's lab. I am currently doing research on electronic skin and fiber interfaces. I am very pleased to be organizing today's seminar. Today's topic is an overview of cancer nanomedicine and will be moderated by Kyudan Park. Kyudan Park, please get started with the seminar.
Good afternoon. This is Kyudan Park from Professor Myung-Chul Choi's lab. I'm currently studying the persistence length of microtubules polymerized with certain drugs, and I will be your moderator today. Cancer is a widespread disease. However, even though researchers are trying to find a cure, 99% of cancer drugs don't even make it to the target cell. By having nanoparticles carry the cancer drugs, we may have a chance to target them directly to the tumor. This is the power of nanomedicine. To further explain about this, we will be presenting an overview of cancer nanomedicine. First, Sujin Kim will explain what nanomedicine is, followed by Umi Ryu, explain how they are designed to fight cancer. Next, the panelists Min-Chul Jang, Han-Jun No, and Junhee Han will each discuss about the pros and cons of nanomedicine along with its future prospects. Now let's start the presentation with Sujin Kim's introduction of what nanomedicine is. Sujin Kim, let's start the presentation.
Well, it's nice to meet you, everyone. I'm Presenter Sujin Kim, and I'm a member of Professor Park's lab. My research area is related to nanomaterial toxicity, and our group will be discussing the topic of cancer nanomedicine today. So first, I will start with the topic of the present and future of nanomedicine. This image is a scene from the movie A Fantastic Boys released in 1966. In it, doctors in submarine downsize to the size of the cell, enter the patients, and navigate through the patient's bloodstream. Eventually, they get into the patient's brain and break into the clogged blood vessel. American physicist and science fiction novelist Dr. Isaac Asimov wrote a very provocative story, knowing that this is entirely impossible physically. 20 years later, in 1986, American scientist Eric Drexler suggested that the product imagined scenario could be realized through a book entitled Engines of Creation. That would be by using nanotechnology. He noted that diseases in the human body usually begin with microscopic change at the nanometer level, and he cited bodies as one of the most promising areas for nanotechnology. 20 years later, from the mid-2000s, nanomedicine's possibility began to expand ever further. In particular, they are being tested in various ways, suggesting a possible solution to treating cancer and infectious disease. The most representative one is the development of a drug delivery system that effectively delivers drug to the disease site in a patient. Nanomedicine refers to a fusion technology that combines medicine with the particle-based technology developed at the molecular level of nanosize, such as diagnosing and treating disease or repairing damaged tissue using nanotechnology. Nanomedicine can be used in various fields such as nanobiosensor, nanoimaging, nanodrug delivery system, nanotissue engineering, and nanodevice. The global nanomedicine market is expected to grow from $14 billion in 2016 to $35 billion in 2025. In this situation, diagnostic and drug delivery systems using multifunctional nanoparticles based on nanotechnology are being actively developed worldwide. Nanotechnology refers to a technology which synthesizes, assembles, and control substance in small-sized units such as atom or molecular result and identify the property of substance. As the size of a given material decreases to the nanometer scale, it is possible to develop useful application methods of various fields using the property of a material while having excellent property differs from those seen at the voltage size. Nanomedicine refers to the use of the physical property of multifunctional nanomaterial at the scale of 100 nanometer of lab in medicine using nanotechnology. Many different types of nanoparticles can be used from a medical and biological perspective. Nanoparticles can be developed in various fields using the difference in intracellular distribution according to the size and physical property and the surface. Therefore, utilize nanotechnology and medical technology or during research, it is essential to grasp nanoparticle physical-chemical property and select appropriate particles appropriately. An example of nanotechnology medical application is a technology that delivers drug or gene for diagnosis and treatment. And this can be applied to a variety of technology in order to detect precision or target disease-specific marker. It can also be used as regeneration engineering and contract agent by using the physical property of nanomaterials. Next, let's look at some of the representative technology being researched in the nanomedicine field. With the development of nanotechnology, drug delivery of specific cells using multifunctional nanoparticles attracts our attention. Based on this research result, nanodrug delivery technology minimizes the potential side effects of drug and can deliver drug more effectively. In the case of common use chemotherapy, there is an issue there where the anti-cancer drug not only attack the cell of the tumor tissue, but also affect the surrounding and other normal organ distanced from the site. Thus, various efforts are being made to solve this side effect by using nanomedical technology. A representative of this is manufacturing nanoparticles at the scale of several nanometers, detecting drug and selectively delivering this nanoparticle to the tumor tissue to be treated. The role of nanoparticle in the drug delivery system using nanoparticle can be compared to that of a delivery bus. The drug contains are in the safely protected until they are delivered correctly to the affected area and are not lost during delivery. And if drug is easily released after delivery, it can increase the effectiveness of treatment and suppress toxicity or side effects imposed on other organ and healthy cells. Most of the carrier bags we use in our daily life have a hexahedral structure of a similar shape. However, to effectively deliver drug in a complex physiological environment, such as the human body, the structure and chemical composition of nanoparticles is being studied in a wide variety. Some don't use biodegradable nanoparticles such as Doxil. Nanoripogen contains doxorubicin and abraxane, and moving nanoparticle containing paclitaxel. These particles cannot selectively treat the target cancer, and they take advantage of the enhanced permeability and retention effect, which passive follow nanoparticle size. A variant nanoparticles such as quantum dot, cold nanoparticle, and dendrimero are being developed as diagnostic and therapeutic agents. Some issues need to be resolved, such as toxicity problem. A reactive particle can be left over due to accumulation in the living body after injection. However, drug delivery technology using nanomaterial can dramatically improve drug efficiency and focus the drug on the actual target. Also, depending on the size of the nanoparticle in question, it is possible to maximize the therapeutic effect by allowing the drug to stay in vivo for the full required time. A recent report by a research team at the University of Toronto in Canada has shown that nanoparticles of different sizes should be used depending on the size of the tumor tissue plus the shape of the surrounding blood vessel. And a research team at the University of San Diego reported that wrapping nanoparticles with the cell membrane of a red blood cell extracted from the patient's own blood could suppress the inflammatory response and improve drug delivery efficiency. The research team at MIT in the United States developed a smart nanomedical system that releases drug only when accurately delivered to cancer cells by making nanocapsules several nanometers in size and device and platform that control the external proteins to destroy cancer cells selectively. As such, nanoparticles do not merely act as a carrier box, but often they are packaged after delivery and can possess artificial intelligence. They show their products will be being actively studied. Nanoparticles possess a relatively large surface area according to their size and the inherent characteristic of the particle, making it easy to synthesize to target materials. And by allowing the synthesis of various substances in one particle, we can increase the accurate target ability. A variety of nanoparticles using these properties are being used in medical applications within the field of nanosensing. Nanoparticles can be applied as a technology to diagnose or detecting via greatly amplifying trace amount of biochemical elements. In addition, in real advantage, varying the amount of sample required for diagnosis can be significantly reduced because it can increase precision and sensitivity and compared to existing diagnosis or taking technology. Furthermore, by using multifunctional nanoparticles, various tests can be performed on one material at once. The test process quick and simple, and the effect of the increasing cost efficiency can be expected. This method combines nanotechnology based on the commonly used ELISA technique. This is an oligonucleotide immunoassay method designed with gold nanoparticles to amplify the fluorescence signal in large quantities of a particle surface instead of generating the fluorescence detection signal via the engine. In nanomedicine technology, nanocontrast agent and nanotarget particle for imaging are actively developed and utilized using nanomolecular imaging technology, which combines nanoparticle with molecular imaging technology. Nanomolecular imaging is a technology developed to perform complex treatment while treating and diagnosing pathogen and etiology in vivo using nano-sized particle or peptides. Stanford University has established an MIPS program and is developing a carbon nanotube-based complex gamma imaging technology which can simultaneously track various biomarkers relative to intractable cancer disease. The University of Frankfurt, Germany, meanwhile is equipped with a facility for the large-scale screening treatment of radioactive peptide therapeutic of neuroendocrine tumor, playing a central role in molecular imaging fusion therapy in Europe. Korean KIST has developed optical imaging nanoparticles which primarily accumulate in cancer cells in mice, while using polymer nanoparticles containing a fluorescent substance. Recently, nanomolecular imaging systems are actively conduct research worldwide to apprehend the actual inflammation of tumor target model. By using nanomedicine technology, we can expect to strengthen the efficacy of a drug and reduce side effects thanks to new drug development. Molecular imaging can bring about time and economic mitigation effects for pathogen distribution as well as drug efficacy analysis. And in terms of development of diagnostic technology, more precise and rapid disease diagnosis will be made possible. It is also expected that the development of nanomedicine will continue to make new contributions in terms of economic and social health, as it can provide many advantages in the academic aspect of understanding the underlying cause and pathology of disease. In order to realize the expectation from nanomedicine, it is necessary to understand the study new technology and concept from a multifaceted and convergent perspective, such as life science, medicine, chemistry, and physics. The ultimate goal of nanomedicine will be the development of kinds of nanorobot that Isaac Asimov and Eric Drexler dreamed about. It would be wrong for a medical nanorobot will make fantastic leaps inside the bloodstream of the body and help to protect our health. Thanks for your attention. And next time, Umi will present about cancer nanomedicine.
Thank you for your presentation, Sujin Kim. Let's now further learn how nanomedicine is designed to treat cancer with Umi Ryu's presentation. Umi Ryu, let's start the presentation.
Hello, I'm the second presenter for this seminar, Umi Ryu. I'm in Bio and Neural Interface Laboratory with Professor Songjun Park. I'm currently focusing on the fiber sensors and fiber medical devices. And today I will talk a little more about the details of nanomedicine for the cancer therapy. Nanomedicine is integration of nanotechnology and the medicine. It includes development of nanoparticles and nanostructured surfaces, as well as analytical techniques for molecular diagnostics, treatment, follow-up, and therapies for the disease. According to WHO, cancer is one of the major serious health concerns worldwide. It is second leading cause of death after heart and vascular disease, accounting for 1 in 6 deaths. 1.9 million new cancer cases were projected to be diagnosed in this year, and 630,000 were projected to die from the cancer this year. Cancer cases are expected to increase by 70% in the next 2 decades, and this is why scientists and the engineers focus on nanomedicine for cancer treatment. Conventionally, cancers are treated with surgical procedures on the site of tumor, followed by radiotherapy and chemotherapy. The current radiotherapy may cause skin irritation, adverse effects such as damage to nearby healthy tissues, and etc. The chemotherapeutics is also limited due to their non-specific effects on healthy cells, the poor pharmacokinetics, and the raised concern on the potential toxic effects. Thus, the nanomedicine emerged as a promising alternative technology, allowing the selective accumulation of systemically administered chemotherapeutics in tumor tissue. The therapeutic efficacy is affected by the circulation time of these nanomedicines in the blood, the nanoparticle protein interaction after administration, and the complex interplay at the perivascular tumor microenvironment due to the presence of multiple biological processes. When nanomedicine is administered into the body, first barrier they face is the biological barriers. Biological barriers provide the primary human defense mechanism, and they have an important function in the drug delivery by blocking the penetration of foreign substances, including engineered nanoparticles, and hindering their access to the target organ. The phagocytic cells recognize and eliminate the foreign objects via mononuclear phagocytic system, which may cause a decrease in the therapeutic efficacy of cancer nanomedicine. Kidney filtration plays another huge role in preventing potential human health hazards, resulting in the clearance of the administrated nanomedicine. When the nanomedicine arrives near the tumor tissue, they are introduced to the tumor microenvironment. Tumor microenvironment is comprised of the extracellular matrix that includes cancer-associated fibroblast cells, endothelial cells, blood vessel-forming cells, and tumor-associated immune cells. As tumor mass initiates the angiogenesis, the blood vessel rapidly proliferates to form disorganized and abnormal vascular systems by overexpressing different angiogenesis factors. The leaky tumor vasculature and poor lymphatic drainage result the enhanced permeability and retention, which is known as EPR effect. Another tumor microenvironment condition is the low level of oxygen partial pressure in the tumor cell, which is called hypoxia. It is well known that hypoxia contributes to the tumor resistance in radiotherapy and to different chemotherapeutics, and also suppress the immune reactivity and promotes tumor angiogenesis and metastasis. Along with these, in tumor tissues, high rate of glycolysis lead to tumor pH vary in range of about 5.5 to 7.0. With all these critical factors encountered, nanomedicines for cancer treatment were tuned by their particular chemical composition and their other specific structural features, including surface properties, general physical characteristics, and functionalizations. Currently, a wide variety of platforms are being investigated for cancer treatment, including lipid-based, polymer-based, inorganic, viral, and drug conjugated nanoparticles. To briefly explain the history of cancer nanomedicine. Liposome structure was first introduced in academia in 1954. After about 30 years, liposomes became the first class of therapeutic nanoparticle to receive clinical approval for cancer treatment in 1995. In early 2000, nanoparticles including polymerase microbes, single RNA polymerase nanoparticles, iron oxide nanoparticles, and etc. were studied, some of which were approved for the clinical trials. There are many more types of nanomedicine with different functions, but in this presentation, I'll introduce 6 major types of nanomedicine. Protein drug conjugated nanoparticles consist of proteins directly conjugated to the drug molecule. They are typically very small. They're very small, like 10 nanometers, to allow particles to have a long half-life in vivo and thus facilitate its drug delivery function to the target tumor site. Liposome-based nanoparticles are nanoparticles created via the use of lipid bilayer, yielding spheres roughly between 50 to 500 nanometers. Since the lipid bilayer is similar in structure to the cell membrane, the liposomal nanoparticles can either fuse with the cell membrane or live once combined with the harsh environment inside the cell. It could release the drug inside the cell. Coating of the surface also lead to key benefits such as increasing circulation time, improving bioavailability of the encapsulated drug, increasing the targeting efficiency, and altering the surface charges of liposomes. Polymerase nanoparticles are comprised of synthetic polymers, allowing customization of many key properties such as molecular weight, biodegradability, and hydrophobicity. The synthesis of polymerase nanoparticles have also been well studied, and a variety of methods were designed to efficiently encapsulate the drug molecules and different drug molecules. Dendrimatic nanoparticles are comprised of dendrimers, which are spherical macromolecules with many branches originating from central point layer by layer. By using specific initiator core, the size and the degree of branching of the dendrimer can be easily manipulated, allowing minimized polydispersity. The hydrogel are the three-dimensional network of cross-linked water-soluble polymers that are able to retain fluid in large quantities. Most synthetic hydrogels are not biodegradable, but enzymatic, hydrolytic, and stimulus-responsive components can be added into the hydrogel matrix in order to create nanoparticles that are degradable under certain conditions. There are many other inorganic nanoparticles in the last category, but such as gold, carbon nanotubes, and silver. Gold nanoparticles are suitable as anti-cancer therapy for its photothermal properties. It also could show the controlled release of the drug by adding a layer of thermal responsive polymers on the surface of nanoparticles. Single-welled carbon nanotubes form highly stable suspensions in physiological buffers, making them suitable for use in biological environments, including tumor microenvironments. For silver nanoparticles, silver reacts with the acidic environment that is often found in the cancer cells to create the reactive oxygen species, which can induce the damage to the cellular material and apoptosis. The last slide of my presentation is the list of US FDA approved nanoparticles for cancer therapy for the clinical trials. And thank you for listening.
Thank you for your presentation, Ovidiu. Next, the panelists will discuss about the advantages and disadvantages of nanomedicine and its future prospects. Let's start the discussion with Min-Chil Jang explaining the advantages of nanomedicine. Min-Chil Jang, let's start the presentation.
Hello, I'm Min-Chil Jang in Master Course in Geoparks Laboratory. I'll introduce you about advantages of cancer nanomedicine. At first, nanomedicine has increased surface area. One feature of nanoparticles that gives them unique physical properties is a large surface area relative to size. As particle size decreases, total surface area increases exponentially. An increase in surface area means that a greater proportion of atoms are located on the particle surface relative to the core. This phenomenon makes nanoparticles more reactive compared with conventional larger molecules. Increased surface area is also responsible for the enhanced water solubility and bioavailability. The large surface area of nanoparticles also allows them to be designed to include a broad range of surface characteristics, including conjugation with electrostatic charges or biomolecules. Next advantage is permeability. If nanoparticles are properly designed, their small size can enable them to cross physiological barriers to deliver drugs to sites that are not normally accessible by traditional means. For example, to reach the lower respiratory tract past the carina, the inhaled particles should be less than 5 micrometers in diameter, especially the particle size with the most efficient deposition in the small airways is said to be less than 2 micrometers. Also, for cancer therapy, enhanced permeability and retention effect can be used for nanomedicine. Enhanced permeability and retention effect, which is called EPR effect, is the property by which particles within the size range of 10 nanometer to 1 micrometer tend to accumulate in tumor tissue much more than they do in normal tissues. Nanomedicine has improved bioavailability. Nanoparticles are generally cleared from circulation by immune system proteins called opsonins, which activate the immune complement system and mark the nanoparticles for destruction by macrophages and other phagocytes. Neutral nanoparticles are opsonized to a lesser extent than charged particles, and hydrophobic particles are cleared from circulation faster than hydrophilic particles. Nanoparticles can therefore be designed to be neutral or conjugated with hydrophilic polymers, such as PEG, to prolong circulation time. The bioavailability of liposomal nanoparticles can also be increased by functionalizing them with a PEG coating in order to avoid uptake by the RES. Liposomes functionalized in this way are called stealth liposomes. Nanoparticles are often covered with a PEG coating as a general means of preventing opsonization, reducing RES uptake, enhancing biocompatibility, and increasing circulation time. Nanoparticles can also be made water soluble if they are coated with a hydrophilic polymer, or they can be made amphiphilic or hydrophobic if they are coated with aliphatic surfactants or liposomes. Lipid coatings can also improve the biocompatibility of other particles. Next advantage of nanomedicine is controlled release. Nanoparticles can be designed so that they can be activated to release therapeutic or diagnostic cargo in response to a site-specific or remote trigger. Properties that can be used to functionalize nanoparticles for controlled release include pH, temperature, magnetic field, enzymatic activity, or other features such as light or radiofrequency signals. Nanoparticles constructed with pH-responsive materials can be designed to trigger drug release at the target site upon detecting a change in pH. For example, Marburg effect is defined as an increase in the rate of glucose uptake and preferential production of lactate even in the presence of oxygen. Cancer tissue shows Marburg effect, and because of lactate, its pH is lower than normal tissue. Nanomedicine can use this feature to make cancer targeting. Thermally responsive linkers consisting of nucleic acids, peptides, proteins, lipids, carbohydrates, or polymers can also be used to attach one or more agents for controlled release from the nanoparticle. Tumor site-specific conditions that could be used to trigger release might include abnormal oxygen levels, unique biomarkers, or exposure to proteolytic enzymes that are overexpressed in tumors. The tunability of nanoparticle properties is an important and powerful concept. Nanoparticles have a broad range of tunable biologic, optical, magnetic, electric, and mechanical features that differ dramatically from the same materials in larger forms because of modified quantum mechanics occurred at the nanoscale level. By changing the size of a nanoparticle, researchers can fine-tune many different properties of nanomaterials. For example, they can achieve different colors of fluorescence by changing the size of a nanoparticle, allowing a means of color coding or labeling during diagnostic imaging applications. Gold nanoparticles' interaction with light is strongly dictated by their environment, size, and physical dimensions. Oscillating electric fields of a light ray propagating near a colloidal nanoparticle interact with the free electrons, causing a concerted oscillation of electron charge that is in resonance with the frequency of visible light. These resonant oscillations are known as surface plasmons. As particle size increases, the wavelengths of surface plasmon resonance-related absorption shifts to longer, rather wavelengths. In conclusion, nanomedicine has increased surface area and can permeate easily to cancer tissue. Also, we can control circulation time, filtration time, and drug release by conjugating nanoparticles, so nanomedicine has advantages in bioavailability and controlled release. Finally, researchers fine-tune many different properties of nanomaterials. Those features make nanomedicine most promising drug for cancer therapy. This is end of my presentation.
Thank you for the explanation, Min Chejang. Next, Hanjun No will discuss about the disadvantages of nanomedicine. Hanjun No, let's start the presentation.
Hello, I'm the second panelist, Hanjun No, and I'm from MC Choi's Biophysics Lab. And my research topic is about physical properties and applications of microtubules, which is a self-assembled protein nanotube found in our cells, especially in axons. So I will talk about the disadvantages of nanomedicine. So the first disadvantage is the problem related with the controllable and reproducible synthesis. And second, the risk of using nanomedicine. And finally, the issue of the financial cost. So first of all, controllable and reproducible synthesis. So the optimal physical chemical parameters, which are size, shape, composition, surface charge, surface corona, and aggregation. So these parameters are crucial for the performance of the nanomedicines. However, usually nanoparticles are formed with high polydispersity, meaning that the variation of the size and shape of nanoparticles are quite large. So unless we establish the precise control over monodispersed and highly reproducible nanoparticles, the efficacy of nanomedicine may be reduced or even cause side effects. And the risk of using nanomedicine. So as Mincher said, nanomedicines are very small and the surface area is increased. So often it results in augmented chemical reactivity. So this can lead to unpredictable interactions and behaviors of nanoparticles in different biological conditions, such as translocating from site of injection to other body parts or undesirably entering the blood-brain barrier. So blood coagulation and the accumulation of nanoparticles in organs, so nanoparticles that do not degrade or degrade slowly, can cause chronic inflammation. And also, the nanoparticles are reported to be related to cause pulmonary diseases or lung injury. So besides these health risks, the disposal of nanomedicine can also cause environmental pollution like air pollution or water pollution. To eliminate or reduce these dangers or factors, evaluation and screening is very important. However, using in vitro models may not capture the intricate interplay of nanoparticles with actual physiological barriers. And also, the lack of tumor models that can recapitulate human cancers is also a big problem because it is the main cause of the discrepancy between the efficacies obtained in preclinical studies and the outcomes from the clinical trials. And about the financial cost. So the scale up of simple nanoparticles, such as liposomes and polymeric systems, can be achieved by using manufacturing unit operations already available. However, if you're trying to make more complex nanomedicines, for example, nanomedicines carrying 2 or more therapeutics, it may require modification of the existing unit operations or development of novel manufacturing processes. So it means lots of monies are required. And also, a group from ETC Group says, the global health crisis doesn't stem from a lack of science innovation or medical technologies. But the big problem is poverty and inequality. So new medical technologies are irrelevant for poor people if they aren't accessible or affordable. So if you see the average cost per dose from the 2009, so doxorubicin is an anti-cancer drug, and Doxil is a nanomedicine to containing the doxorubicin. You can see that the doxil is much more expensive than the doxorubicin. And again, similar in the case of paclitaxel, which is another anti-cancer drug. So the nanoformulation of paclitaxel is way more expensive than the usual paclitaxel. And the big business of nanomedicines had an estimated value of $53 billion in 2009, and the industry is predicted to reach a total market value of approximately $334 billion by 2025. So regarding the fact that most of the countries still today are deprived of the basic medical facilities, so these kind of imbalanced investments on the advanced nanotechnology, such as nanomedicine, it can be questioned. So in conclusion, nanomedicine is indeed an emerging research area in medical nanotechnology, and it has several strengths and potentials. However, for the further development of more complex and high quality nanomedicines, so numerous challenges still remain, such as toxicity, reproducibility, and cost. So these are the references, and thank you for your attention.
Thanks for the discussion. Lastly, Junhee Han will discuss about the future prospects of nanomedicine. Junhee Han, let's start the presentation.
Hi, my name is Junhee, a professor at BiS987, and I'm the rest panelist for discussing about the limitations and future aspects of cancer nanomedicine. Until now, we explained the conventional concepts of cancer nanomedicine and its application to immune-oncology field. And actually, as shown in the historical timeline of major developments in the field of cancer nanomedicine, concept of using nanoparticles to treat cancer was widely studied in late 1900s. And in 1995, the first FDA-approved nanomedicine Doxil was emerged. And after commercialization of Doxil, many other strategies for developing cancer nanomedicine in specific manner was kept studied. However, as shown in the light table that summarized the currently FDA-approved nanomedicine for oncology indications, there are not so much commercial nanomedicines since studies about them are over 30 years. And as mentioned before, there are lots of hurdles for clinical translation of nanomedicines, and we'll talk about some points for overcoming those limitations. Since the determination of the optimal physiological parameters is crucial for the successful development of therapeutic nanomedicines, controllable synthesis and scalable manufacturing is one of the most difficult hurdles for clinical translation. And many studies suggested the possible solutions for controllable and scalable synthesis. And one of them is using microfluidic technologies that allows high-speed self-assembly of polymeric nanoparticles with narrower size distribution and tunable physical-chemical properties and greater batch-to-batch reproducibility. And also similarly, particle application in not wetting template print technology has enabled the synthesis of monodispersed nanoparticles with precise control over size, shape, chemical composition, and drug loading properties, which allows the reproducible fabrication of nanoparticles. And also both effective in vitro and in vivo therapeutic evaluation and screening methods of nanomedicine before translating to human studies are big hurdles. Since conventional in vitro models using cultured cells in multi-way plates lack the complexity of biological tissues, organ on a chip and tumor on a chip models are suggested as their substitutions. And those organ or tumor on a chips can well reconstitute the tissue-tissue interfaces that allows various real-time analysis of nanoparticles at physiological flow conditions, so thereby expanding the capabilities of cell culture models. And finally, for in vivo evaluations, diverse animal models are currently available, including cell line-based subcutaneous and orthotopic xenografts, and patient-derived xenografts, and genetically engineered mouse models. However, no single model can fully reproduce all aspects of human malignancy, and EPR is generally more consistent in animal models rather than in cancer patients. Furthermore, considering the major contribution of tumor metastasis to cancer mortality, models of human tumor metastasis will be invaluable for the evaluation of APR and nanoparticle penetration and targeting in metastatic tissues compared with primary tumors. Therefore, the translation of nanomedicines will be greatly improved by the development of animal models that mimic closely the heterogeneity and anatomical histology of human tumors, such as high-fidelity PDXs and humanized mouse models, and genetically engineered mouse models with aggressive metastasis. Besides the challenges for clinical translation, cancer nanomedicine has met new era with growth of immunotherapy. And beginning approximately 10 years ago, clinical trials showed significant efficacy of checkpoint blockade and chimeric antigen receptor T-cell therapy in certain cancers, and leading to approval by the US FDA of multiple therapies. And the paradigm shifts of oncology towards immunotherapy occurred. And those immunotherapy offered new opportunities for cancer nanomedicine in many ways. Conventional cancer nanomedicines that delivered chemotherapeutic agents met challenges, include the fact that by current technologies, they deliver only a small percentage of injected dose into the tumor, and some clinical evidences suggested that the efficiency of EPR-based targeting differs significantly among patients. However, nanomedicines aimed at enhancing immunotherapy often have a different set of delivery requirements than systems delivering chemotherapies, and also they can act by a number of mechanisms distinct from chemotherapy. Because the immune system acts as a highly dynamic network, effective stimulation of a small number of leukocytes in tumors or lymphoid organs can lead to large changes in tumor microenvironment, driven by a cascade of cell-to-cell communication. Also, assistance of nanomedicine allow novel mechanisms of action for immunotherapeutic agents, including the ability to display ligands to immune cells and regulate intracellular delivery of cell-impermeable compounds and control the timing of drug release and activation. And based on those concepts, enhancing the cancer immunotherapy with nanomedicine as a tool are actively going on researches and rapidly commencing the clinical trials. And I will introduce some of them at the frontmost line of development as my final slide. Moderna, that you recently heard of, COVID-19 vaccine, and German company BioNTech are leading companies that exploit simple nanomedicine formulation to deliver mRNAs in order to enhance the cancer immunotherapy. Moderna, founded from Professor Robert Langer laboratory, developed stable formulation of lipid nanoparticles for delivering mRNAs that encodes the molecules that can activate both antigen-presenting cells and T cells. And this concept of nanomedicine is now ongoing clinical trials. BioNTech, which also founded from Professor Sahin Group, developed mRNA lipoplex formulation that can specifically target the antigen-presenting cells at lymphoid organs simply by adjusting the size and the surface charge of liposomes. And those specific delivery to APCs in lymphoid organs enabled antigen-specific immune responses and also exploited for activating CAR T cells against solid tumors. And likewise, not quite highly engineered but simple concept of nanomedicines are widely exploited in the field of cancer nanomedicine and in the future. And this is the end of my presentation, and our moderator will finish up our presentation.
Thank you for the last presentation, Chunian. Now we will have a Q&A session. If you have any questions, you can type your question on the chat board and we'll view them one by one. And there seems to be a question already. It's from Anna Pavlovna. First, thank you for a nice presentation. I have two questions. Do you think that the expensive price for the nanoparticle drugs is reasonable considering the research and producing costs? Is there even some solution how to reduce the price of these drugs? Thank you for your answer. Is there anyone who would want to answer this?
Maybe I should answer the question. So actually, once the manufacturing system or facility is established for a new developed drug, so the price will eventually go down as time goes on. So I think that is kind of same for all the drugs. So once the drugs are developed, initial stage, the price it is unavoidable. The price will be very expensive because lots of investments will be made for the scaling up process from the laboratories to the industry. So maybe like I don't know if there's some solution for reducing the price even from the initial stage. I don't know, but eventually the price will go down. But I mean like, yeah, you know.
So probably should wait for a while to see if there are any more questions. Well, if there aren't any, well, we could end the seminar here. And I would like to thank all the people who organized and gave the presentation. Thank you everyone for your attention and see you next week.
Okay, see you next week.